Blessed day everyone, this is Richard Devera again. Our topic today is about mutation and gene regulation. This topic is about why there is a genetic disorder, and we wonder why there is a member of our family that there is a disease like this because of what we call the responsible mutation. And the gene regulation, this is the element to regulate the transcription and to modulate translation of mRNA. This is the act to induce or repress the expression of a gene. Mutations are changes in the genetic sequence, and they are a main cause of diversity among organisms. And mutations can be inherited. Parents that have mutation can pass them to their offspring. And germline mutations are present in every cell of an individual, including the egg or sperm used in the production of offspring. Renewable mutations means new, occurs by chance in one of a few eggs or sperm or just after fertilization and are not present in every cell of a parent. This explains situations where a child has a genetic disorder that is unseen in the family history. And some mutations are acquired in environmental agents called mutagens, can alter DNA. An example of a common mutagen are the UV wavelengths in sunlight associated with skin cancer. Acquired mutations are typically not passed to offspring but can be if there are altered DNA sequence in egg or sperm. There are types of mutations. The first types of mutations is the point mutations, a change in a single nucleotide. Deletion changes the number of DNA bases by removing a piece of DNA. Insertion, one or most bases are inserted into the original DNA base sequence. This is sometimes called a frame shift mutation because it shifts the codon reading frames. Duplication consists of a piece of DNA that is abnormally copied one or more times. This type of mutations may alter the function of the resulting proteins. Inversion is a type of mutation where a sequence of nucleotides in the DNA is reversed. And that's it, the mutations. Now we are going to in gene regulation. Gene regulation includes a wide range of mechanisms that are used by cells to increase or decrease the production of a specific gene product, that is the proteins or RNA. Gene regulation can occur at various steps. The amount of product depends on: number one, the rate of mRNA synthesis. That's involved the transcription. It is the first step in gene regulation. It involves copying a gene's DNA sequence to make an RNA molecule. And the second is the mRNA degradation. Its mRNA differs in the rate at which they are translated into protein and in the stability of the mRNA molecule. The longer lasting an mRNA molecule is, the more protein products that can be transcribed from the mRNA sequence. Number 3 is the protein synthesis. The translation process is involved here, by which a protein is synthesized from the information contained in a molecule of messenger RNA. Then a transfer RNA, tRNA, the termination of RNA, molecule carrying the amino acids methionine binds to what is called the start codon of the mRNA sequence. And some examples of gene regulations are: enzyme induction is a process in which molecules, like drug, induces initiates or enhances the expression of an enzyme. The lack operon is an interesting example of how gene expression can be regulated. The viruses, despite having only a few genes, possess mechanisms to regulate their gene expression, typically into an early and late phase, using collinear system regulated by anti-terminators or splicing modulators. And that's it, everyone. That is our topic about the gene regulation and mutations. I hope you learned something to our topic. Have a blessed day, everyone, and thank you.